All right, welcome biology students to Hanson Studios. This is my little shop in the backyard. I found a nice way to comfortably do this, and against my original intentions of doing uh, YouTube videos, I think it's going to be more helpful to do. Well, they might still be able to be on YouTube, but I'm using Zoom as my platform. We're still going to meet Zoom on at the Zoom times of the Zoom schedule that the RAA admin put out, and I'm going to be there at those times for questions and answers, worship thoughts, things like that. But I won't be lecturing during those 30 minutes. I'm going to be creating these videos and sending them to you that you don't have to join, but you can watch on your own time. I found out Zoom actually is pretty nice for creating videos. I'm able to do a lot of the things that I wanted to do without all of the equipment and headache that I tried. So I finally got this figured out. Thank you for being patient for this first video. Today's video, this one's going to be about um, RNA, what is RNA, how it's different from DNA, as well as how is it made. Okay, this is the first section in chapter 13. So we're going over chapter 13, section 1, the first section. This whole video is going to be about the first section. When we're done with that, you can go ahead and probably be well prepared for doing the assessment at the end of the section for homework. So before we do that, let's kind of recap what DNA was. Okay, we're about to be talking about RNA, which probably is very foreign to you. Everyone talks about DNA. DNA gets all this credit, right? Because DNA is like the molecule, right? We lift up DNA all the time, but it's actually RNA that does all the work. Now, of course, without DNA, you're not going to have any RNA because it takes DNA to make RNA. You make RNA by interpreting DNA, but when it comes to doing all the dirty work and making the proteins, it's the RNA that does that. And you're going to see in this chapter how that happens, and you're going to appreciate probably RNA more than you do DNA. But let's recap again, like the function of DNA. And just want, before I go into the PowerPoint and slides, I wanted to just use the analogy we used in class a lot of the book. So DNA is like a book, right? It has a message inside of it. Just like this book has a message. Now, if you don't know how to understand the sequence of characters, this book doesn't make any sense to you, right? But because I can translate the sequence of characters in this book, I can figure out, okay, these characters together make the word uh, protein, whatever. These characters make together the word home. Um, and so I can then formulate those words to make a sentence and so on, and make paragraphs and all kinds of thoughts. And so you can get complex with writing. Uh, and DNA is like a book. It has characters, which you learned in the last chapter, come in the form of nitrogenous bases. And the specific sequence of those nitrogenous bases is important for what protein is going to be made, and the genes that you have on your DNA. So, but again, I just wanted to use this analogy, and a good way to think about this is that the book is like your DNA. This is the original, right? Let's suppose that this was the only book we had for, this is a book I own for biochemistry. And let's say that this was the only book I had, and I needed to make photocopies for all my students. Well, just like the cell would need to copy its DNA to make RNA. Um, Basically the cell is going to hold on to this DNA inside of the nucleus. The DNA never leaves the nucleus, and so let's imagine that I have I have my laptop bag here that I was going to use. Let's say that I put the book inside the laptop bag and I zip it up. There. Now inside the laptop bag is the book. If we pretend the laptop bag is the nucleus, then just analogous to the cell having the DNA in the nucleus, we have a bag holding the book. The book is, can't turn this upside down, is not, I zipped it up, it's not going out, right? Now this is what's kind of neat. In order for me to make photocopies, I would probably have to take this out and run into a photocopy machine. But imagine there being a photocopy machine inside of this bag. And then when I needed photocopies, I could just unzip it and pull out photocopies and leave the DNA inside, leave the original copy inside. That's actually what happens with we call the process transcription, and that's what we're going to go over today. And all that happens inside the nucleus of the cell, inside the nucleus of the cell. Then the RNA can be taken out into the cytoplasm and be translated. But let's go into the PowerPoint. Let me connect here so you guys can see what's going on. Oh, I first need to, sorry, set this up. Share. Okay. Connect. All right. Boom. Nice. I love this. Okay. Let me drag this over here so you can hopefully still see me. Okay. Here we are. We got two key questions in this section. What does RNA, no, I'm sorry, how does RNA differ from DNA, right? You've done a lot of talk about DNA, but what is different about RNA? And there's actually three main differences we're going to go over there. And the second question is, how does the cell make RNA? How does the cell make RNA? I believe the last chapter, you learned how DNA copies itself, so you know how DNA does that using the base pairing rules and enzymes called DNA polymerase. But how do we make RNA? You'll see it's actually pretty similar using the same concepts and techniques. So let's dive right in. If you look here, let me go ahead and get this going here so you can see this better. I'll use my pointer. You can see here's our key question. How does RNA differ from DNA? Well, let's just jump the gun and scroll down here to the key sentence that answers the key question up here, right? You guys hopefully can do this reading on your own, but I want to point out the major differences here. And I also have some images I want to show you that the textbook does not show, which I think are pretty important and should be seen. So number one, there are three main differences between RNA and DNA, but number one, the sugar in DNA, the sugar in RNA is ribose instead of deoxyribose. So that's why it's called RNA. Instead of DNA, which stands for deoxyribose nucleic acid, RNA stands for ribose nucleic acid because the sugar is ribose instead of deoxyribose. But it's still nucleic acid. Remember at the chapter 2 at the beginning of the year, we talked about the 4 macromolecules of life, proteins, lipids, and carbohydrates, and then the fourth one was nucleic acids. And so this is a major molecule. This is a huge molecule, gigantic polymer made of thousands, millions of nucleotides long. And so this is a huge molecule. And in that molecule, in each little nucleotide that you want to dissect, if you want to look at each nucleotide individually, you'll see that the sugar is ribose instead of deoxyribose. And literally, that just means the difference between an oxygen. One has an oxygen and one doesn't. Well, they both have oxygen, but one's missing an oxygen that the other one has. Right? Small difference, but nonetheless, it is a difference. Okay. The second point of notion is here is that, and I don't see it connecting here. I'm kind of worried about this on my screen. Oh, there it is. Okay, here. Number two, RNA is generally single-stranded and not double-stranded. So DNA is the fancy double helix, meaning that there's 2 strands of nucleic acid that are weakly bonded together by what are known as hydrogen bonds. Hopefully all this is familiar to you. And we don't have hydrogen bond, well, we don't have 2 strands of nucleic acid in RNA because we only have 1 strand. So we don't have 2 strands that complement each other with base pairing and all of that. You literally just have 1 strand of nucleic acid. All right, so that's the second point. The third point here is that RNA contains uracil instead of thymine. So anywhere you would normally see thymine as the nitrogenous base in DNA, replace that with uracil. Very similar looking too. All right, the nitrogenous base kind of even looks the same as far as its chemical arrangement, but it's slightly different. So uracil is a little bit different than thymine, and you just need to know that RNA contains uracil instead of thymine, making it different, making the cell be able to tell the difference between DNA and RNA. All right, and so we want to look at the role. Let's look at the roles of DNA. And I love this image up here at the top. This is an awesome visual analogy. Do you see the guy on the left over here with the master plan? Notice he's not giving that out to all the architects and the electricians and the plumbers and the framers. No, he's keeping the master plans himself, and he's making a photocopy, a photocopy, a photocopy, and giving photocopies, the blueprints. He's keeping the white prints. He's making blueprints to give to all of the individual workers that will do stuff. Notice this guy's not even doing anything. He's probably making the most money too. But he's not even doing anything with the plans. You know, he wrote the plans or has the plans, and he's just in charge of making sure everybody gets a copy. He's making sure these individuals get a copy. Notice this guy, and this guy are the same. So this guy is literally in charge of making the copy and distributing the copy to these people. And then these people will take those blueprints, and they, again, depending on what their job is, they'll do different things. I could have the same blueprint you have, but depending on what page I'm looking at or what I'm doing in that room, I would be doing something different than what you would be doing. You know, we'd be working side by side in the room, but maybe I'm running the wires and maybe you're running the pipes for plumbing. So this awesome visual analogy of how RNA works. Basically, all of these blueprints are like RNA, but the master plan, that's your DNA. That's your original copy. And so as far as the functions of RNA go, the functions of RNA, it depends on what type of RNA. So check this out. You've actually got 3 different types of RNA. All right, you've got 1, 2, 3 different types of RNA. You have messenger RNA, which sometimes we just call mRNA. So notice you'll see here, you have mRNA. Sorry, that looks bad. Let me take that off. I'll just use my pointer. You have mRNA. For ribosomal RNA, we abbreviate that rRNA, lowercase r, and then all caps RNA. For transfer RNA, it's abbreviated tRNA. The little t stands for transfer RNA. So again, you have messenger RNA, ribosomal RNA, and transfer RNA. So what are the differences between these 3 types of RNA? Well, I'm a visual person, so let's look over here at these images, right? Messenger RNA is literally just the single strand like this. It just looks like a single strand. Notice it's got the nucleotides there and the nitrogenous bases poking up. The messenger RNA carries instructions for polypeptide synthesis. Polypeptide is a fancy word for saying protein, kind of the immature stages of a protein. And the messenger RNA carries the instructions for that polypeptide synthesis from the nucleus to the ribosomes in the cytoplasm. If you guys want to rewind back to chapter 7, we talked about ribosomes and how they're the factories for protein, or their function was to make proteins. Well, now we're going to look at how ribosomes actually do that. Okay, and so but before ribosomes can make a protein, they need the blueprint, right? So they need this blueprint here. The messenger RNA is like the blueprints you see up here. All right, and again, that gets taken out of the bag, out of the nucleus, and into the cytoplasm to do that. Well, when it gets into the cytoplasm and meets that, what I just said, ribosome, to be interpreted, that's where you're going to run into ribosomal RNA. Guess what a ribosome is made of? Ribosomal RNA. That's the second one down here. Now, ribosomal RNA is actually only part of the ribosome. So like this whole structure here is the ribosome. There's actually 2 subunits, and we're not going to get into all that detail, but you just need to know that this entire structure is a ribosome, and that it's made up of not only ribosomal RNA, but a whole lot of proteins within it. Now, quick side tangent, we're talking about how to make proteins. We're talking about the key players here in making proteins, but yet one of the key players is made of proteins. Which came first, the chicken or the egg? Something to think about. So the ribosomal RNA is a lot like messenger RNA in its structure. It's single-stranded, has the sugar phosphate backbone with the nucleotides just like this, but the way it folds itself together and wraps around the proteins and its function makes it different. It makes it different. So the function of ribosomal RNA is that it forms an important part of both subunits of the ribosome. It basically makes up the ribosome. That's the function of ribosomal RNA. It makes up the ribosome. Your next question would probably be, what's the function of a ribosome? Well, we just said it. It's that it makes proteins. It makes proteins. All of this is going to culminate in the production of a protein. Finally, the third key player here is transfer RNA. So what is the job of transfer RNA? Well, it's going to carry what we call amino acids to the ribosome and match them to the coded mRNA message. And they're perhaps my favorite type of RNA because they have such a cool job of recognizing the sequence of nucleotides on messenger RNA and matching up with them with these sequences of nucleotides, and then also bringing in amino acid. Like, they're the ones that do, to me, like a lot of the work. I love transfer RNA, and it has a really cool look to it. And we're going to look at better images of transfer RNA in just a second. But be familiar with these three roles of the messenger RNA, which is sometimes called mRNA, ribosomal RNA, which is sometimes called rRNA, transfer RNA, which is sometimes called tRNA. All right, moving on. Here's some RNA structure, which I thought you should get. Okay, the book has usually this structure here drawn, but what does this green line represent? It's just a solid green line, but there's way more to the story there. That green line represents a sugar phosphate backbone, which basically makes up the structure, the backbone, right? Kind of the main structure, and it doesn't change from nucleotide to nucleotide. Remember that this is a nucleotide, and this is a nucleotide, and this is a nucleotide, and that this is a nucleotide. Those nucleotides are the monomers that make up the polymer, nucleic acid. But if you want to look at the monomers, the individual units that are like repeating, it's these guys here, these nucleotides, there, and there. All right, let me undo all that because it looks bad. And each nucleotide has some similarities, and there's only small differences. And for each nucleotide, the phosphate and the ribose sugar here, this is the ribose sugar, this is the phosphate, ribose sugar, phosphate, ribose sugar, phosphate, ribose sugar, phosphate. You just get this alternating pattern where the phosphate of one nucleotide bonds to the sugar of the next nucleotide, which obviously bonds to the phosphate of its own nucleotide. And then that phosphate, if we continue this structure on, because RNA is way more than 4 nucleotides long, if we continued on, this phosphate would bond to the next sugar of the next nucleotide. And that sugar phosphate backbone is consistent. Doesn't matter what nucleotide you have. The thing that makes nucleotides different is the nitrogenous base that you have here, or here, or here, or here. And you've already learned about those 4. Remember, you had cytosine, guanine, adenine, and we used to have thymine, but this time for RNA, we're going to have uracil. So always just get rid of T, and start replacing it with U. All right, they're different. It's one of the things that makes RNA different. And so here's a cool image I found too, showing DNA here. This is the double helix, right? Where remember, had the base pairing. The C pairs with G, A always pairs with T, A pairs with T, G pairs with C, or vice versa. C pairs with G. It's called base pairing. Base pairing, and that's what holds the double helix together. Those are weak bonds, by the way. They're just hydrogen bonds, and that's important because we need to unzip the DNA sometimes. Many times we need to unzip the DNA to read it, just like you need to open a book to read it. Well, the RNA doesn't have those base pairing because you literally just have the sugar phosphate backbone with individual nucleotides hanging off. And anywhere you would see a T, anywhere you would see a T, you're now going to have a U, right? All right, this strand here is the same as basically this strand. You can see the C, C, G, G, A, T, but now U for RNA, right? And here's what the nitrogenous bases actually look like. They look kind of cool. Everywhere you see the lines come together, like there, any corner or vertex, those are carbon atoms. So we're not going to get into all that kind of details. That's something you would learn in organic chemistry or biochemistry if you're into that. but those are the chemical structures. And you can keep moving, zooming in, and getting further and further and more detailed. And since I know we're not studying, like I said, each individual nitrogenous base and how they look different and such, I do want you to know that they have this chemical structure here so that it's not super abstract. But we're going to now go back to this more simplified drawing the rest of our talk. Throughout the textbook, we're going to be looking at this simplified drawing. And basically what I need you to do is remember this base pairing, because that, even though that's what's used to hold DNA together, we're still going to use base pairing rules to make RNA, because we make RNA from DNA, all right? So stay tuned for that. Let's now go to the next image here. Wait, make sure I didn't go too far. Okay, RNA synthesis. How does that happen? We just said that. Let's jump right into that now. RNA synthesis. RNA synthesis is a process we actually call transcription. That's a vocab word for you there. Transcription, just like you would transcribe a letter or transcribe a text message or an email nowadays. Transcribing means to write something, right? And in transcription, if you just Google the word transcription, I guarantee you're going to get nothing but RNA stuff, because that's what literally, it's a vocab word. Transcription means it's the process where segments of DNA, I'm reading the bold-faced sentence here, that segments of DNA serve as templates to produce complementary RNA molecules. Okay, we got a couple words we need to mention there to discuss. In transcription, segments of DNA, so small segments of DNA here that you see, right? This is your DNA double helix. Small sections of DNA will serve as templates. What is a template? Well, a template is just something you would use to help you write. It's like a guide for you to help you write the right thing, right? And in this case, the right sequence of nitrogenous bases, or the right sequence of nucleotides. We don't want to just slap together a bunch of nucleotides randomly, or it won't make any sense. Okay, and so during transcription, DNA is going to serve as a template. In fact, only one strand is going to do that, and we call that the template strand. The template strand is going to serve as the template for us to make our RNA strand. And guess how we're going to do that? Well, we're going to obviously use this. We have to use an enzyme, which is a large protein that has the function of doing a certain job and creating this RNA strand. Again, that enzyme is called RNA polymerase. But what does that RNA polymerase do? You guessed it, it uses base pairing rules. So you need to be able to just roll that right off your tongue, or commit that to memory. When you see C, it base pairs with G. When you see A, it base pairs with T. But in this case, we're replacing thiamine with uracil. Right? And so if you know how to do those rules, and it's fascinating that it takes me a couple hours, a few lectures to talk about this and help you guys understand how RNA polymerase generates this specific sequence of nitrogenous bases by reading the DNA strand, while meanwhile, in me just finishing that sentence, I don't even know the number of cells that literally just created who knows how many RNA sequences. I'll have to come up with the numbers. I don't want to lie to you. So, but they're astronomical numbers. Your cell does this just like in milliseconds. It does this. It's phenomenal that it has the ability to do this like so quickly. And so, literally one nucleotide at a time. There are thousands of nucleotides along the strand of DNA that this RNA polymerase has to read. Every time it reads a nucleotide, it adds the complementary base. That's what that word complementary means. That word complementary means that it refers to the rules of base pairing, which you learned in the last chapter. And so, again, this whole process is called transcription. The next process, which is going to be in the next section, is called translation. That's how we're going to read it. But we're just focusing on how do we make the RNA right now. And so that process is called transcription. And then you have your RNA strand. All right. Now that, again, the only way that can be done is if number one, the DNA is opened up. So you actually need this enzyme to be able to open the DNA up and to add nucleotides one by one, not necessarily adding it to the DNA, because otherwise then you'd have a strand stuck to the DNA, right? You don't want that. You just want to, RNA is pretty neat in how it's able to use the base pairing rules, but still spit out a strand all by itself. And then there's another enzyme that um, zips back the DNA and puts it back together. So let me show you what I mean by that. And actually, here's a cool image first that just kind of shows you that process just with letters, because you're going to end up doing this a lot, like on the test. And again, this is what like your body's doing. Your cells are doing this constantly. They're reading this. They're reading this template strand. In this case, this would be the template strand. They're going TACTAGAGCATT. And so the RNA polymerase is reading this template strand, and it's going, okay, T, I need an A, I need a U, remember, not T. C, I need a G. T, I need a I need a U. G, I need a C. A, I need a U. And it's making this RNA strand. And then this will get zipped back up. The DNA will get zipped back up together to be the helix form again. And this transcript will be written. This is your transcript. This is your messenger RNA. That's that first type of RNA we talked about. This can now be taken out into the cytoplasm to be translated into the polypeptide sequence, which we'll go over later. But sometimes or not sometimes, the strand that is being interpreted here, the strand that's being interpreted to make this RNA strand here in red, this strand of the DNA is called the template strand because it serves as the template. And RNA polymerase, this kind of bubble here, which is used to represent the enzyme that does that, this key player, is actually reading it in a certain direction, 5' to 3'. It's making the RNA in the 5' to 3' direction. And that has to do with the carbon numbers, and don't want to get into all that, right? We got enough on our plate with this process that we don't need to worry about necessarily the 5' to 3' yet. But here's another interesting point I wanted to bring up that, or kind of just still meditate on here a little bit, that DNA has to be unwound here. DNA has to be unwound. It has to be opened up in order for RNA polymerase, this yellow blob here, to read the template strand. In order for RNA polymerase to read this template strand, the DNA has to be unwound and opened up by DNA helicase. And when RNA polymerase is done, when it's done doing the base pairing like this, then this RNA strand, this messenger RNA, or this RNA transcript will be sent out or sent off by itself, a single strand. But then the DNA has to be rewound back together. It has to be recoiled back together. So keep that in mind. We don't want the DNA unwound if it's not being interpreted. Okay, if you're not reading the book, close it. All right, keep going to that book analogy. All right, finally, let's finish this up here. The last page of this section, and then you guys can answer the 13.1 assessment. Let's, don't worry about number 3 there. Cancel number 3 there. I'm just happy if you just do number 1 and 2. So let's look at, actually, if you want to do number 3, do number 3 for extra credit. Do number 3 for extra credit. I normally don't do extra credit, but that does look like a pretty cool problem. And if you're willing to tackle that, I'll give you some points extra credit. So let's finish this now. And there's some sections of RNA, or sections of DNA, I should say, that are pretty important, that aren't necessarily, I guess, important, but they're unimportant enough to be important. And let me start with this image first, but then I'll go back to the image in the book. I think this one makes more sense. So here's our DNA, right? How does DNA, or I'm sorry, how does RNA polymerase know where on the DNA to start? Because DNA is pretty long. You've got tons of genes all along your DNA. Some of them are for different traits. That was all. Remember when we talked about genetics? How you have genes that code for different traits. Now, really, these genes are going to code for proteins, actually, but those proteins will inevitably make your traits. But my point is, how does RNA polymerase along the huge molecule of DNA know where to start and stop? Well, there's sections of highlighted DNA here that are certain sequences that are called promoters. When RNA polymerase sees that, when it sees that certain sequence, it knows, okay, here it is. Here's where I need to open up the DNA and start adding nitrogenous bases or adding nucleotides to make my mRNA strand. And it just continues on. It just keeps reading the template strand. And as it reads it, remember, it keeps adding the complementary bases, not necessarily adding the complementary bases to the DNA, but adding the complementary bases to the nucleotide before it, so it makes this long polymer. We're making you can see the purple enzyme here, RNA polymerase, making this mRNA strand here. It's making our mRNA strand, which again will be taken out of the nucleus to be translated by a ribosome later. But this promoter is pretty neat. That's a section of DNA that some might say is pointless because it doesn't really code for making any mRNA, but it does tell the RNA polymerase where to start. So I think that's very important. So you have promoters here. That's a vocab word, by the way. That's why I brought you that image. And that doesn't even show up here. So imagine trying to just imagine that. We do have an image here describing what introns and exons are, though. Let's look at that real quick. So RNA also does a little bit of editing, right? You guys have ever edited videos where you add chunks of video or take out chunks of video? Usually taking out chunks of video. Well, that's what RNA will actually do. Now, this is going to sound really weird. I apologize for whoever named this, but sections of DNA that get taken out, like you see this little piece here and this little piece here, they get taken out. They get taken out. Those are actually called introns. No, it sounds weird. You would think, well, if it's getting taken out, wouldn't it be called like the exon or something like that? Is this like exiting? No, it's like backwards in my mind. I feel like it is. Introns will not be interpreted. They just get cut out. They get edited out. They're just not. There are certain sequence that aren't relevant to the translation process. You ever had to write a paper or something like that to your teacher, and your teacher just crosses out a whole sentence, like you don't need that sentence? Imagine like that's what the editing is happening here. There's also, but all of the pieces that do get interpreted, the dark blue, we call those exons. Those are exons. Those are segments of DNA that will get interpreted. And so if you consider all of it, this would be your pre-mRNA, if you have it all. But once you take these introns out, then you have your mRNA, which also has a cap and a tail, just so that your body, your cell can recognize, where's the beginning and where's the end, right? In a nutshell. I don't go into too much detail there, but it does have a cap and a tail, which is just a small segment, a certain sequence of nucleotides that's very consistent, that represents the cap, a certain sequence of nucleotides that represent the tail. And so guess what? Wouldn't it be a problem if you had that same sequence of nucleotides anywhere in this part? It would, right? It would. It'd be like writing in the middle of your essay, please stop reading this essay. Well, that's essentially what would happen if you if you had a sequence in the middle of what needed to be read that said stop here. Like that's pretty much what the tail says and what the cap says. Stop there. All right. So, the text might bring up a few extra things to answer these questions. If you need help with those, join me in the Q&A or whatever, and you can email me. But it looks like these are pretty self-explanatory questions. All right, we did 13.1. Great. So now you know what RNA is, and you specifically know how we made. Let's go back here. You know how we made this first one, messenger RNA. But remember, there's two other types of RNA, ribosomal and transfer. Ribosomal RNA and transfer RNA are two huge players in the game of making a protein, and they're going to come into play in the cytoplasm of the cell, outside of the nucleus. In the cytoplasm of the cell, when we go to make the protein, when we actually translate when we actually interpret or read the messenger RNA and make the protein, these are going to be the two key players that do that. They're all key players because messenger RNA carries the message, but then these two players serve as the host and the active participant in putting together the amino acids to make the polypeptide, which will eventually be folded into a protein. Uh, but and that's going to take a lot of work. We might actually spend 2 videos on translation. Transcription is a little easier. As long as you understand base pairing and kind of how that concept works, then you did just fine. You should understand this just fine. Let me log out here, unmirror here. So, oh, actually, I go up here to stop share. All right. Let's see what our time frame is. Oh, I don't even see it there, but I think we did pretty good. So key takeaways are: can we answer our original key questions? How does RNA differ from DNA? Remember those three points. RNA is single-stranded. It has ribose instead of deoxyribose, and it has uracil instead of thymine. And then how do we make it? We use base pairing rules and a special enzyme called RNA polymerase to read the template strand of the DNA and create a messenger RNA strand. And that can happen over and over and over, just like I can keep photocopying the same pages of a book over and over and over, right? And that's what's kind of cool about DNA is it can stay intact, and the RNA is pretty cool too because it's like indestructible. I mean, it's destructible. It's like, if you mess it up, go make a new one, right? Your body can make thousands and keep making RNAs until finally the DNA isn't good for that anymore, and at that point you just make a new cell with new DNA that's fresh. Okay, sorry about the tangents. Enjoy your homework. See you. Bye.